Hi, I'm Mr. Bradford, and the goal here is simple. By the end, the hard things feel obvious. Today, I'm going to talk to you about two things: code dominance and pedigree charts. So, let's get started. Code dominance, the typical examples that we look at are blood grouping, and then like an example of like speckled hens. So here, for example, you've got your phenotype, black, speckled, and white, and there's two variations of the gene. So remember, a variation of a gene is called an allele. So in this instance here, they've been denoted. You have like the big C, which is showing us that it's codominant, and then followed by a little letter. In this case, B or W. So because we're looking at the coloration, that's why it's been given a C. And here you can see I'll make it a bit bigger. Then this one is homozygous dominant for the or homozygous for the black gene. This one is homozygous for the white, and then but together you get this codominance, which means both of them are expressed. Not one is dominant or one is recessive; they are expressed together. And you, in this case here, we get this pattern. There's something called incomplete dominance. As a side note, where you get like an intermediary. So if it was like one's black, one's white, the intermediary would be gray. So both of them are being expressed, but in an incomplete fashion. So that's incomplete dominance. Similar to codominance, but where you get like an intermediary. Whereas with codominance, both of them are being expressed. The other classic example that we always look at is blood grouping. So you'll probably be familiar with the basic ABO grouping systems: A, B, AB, and O. And here we can see, like, if I show you this diagram here, then what we want to think about, and these are the tables that we need to know them. We need to know that type A means that they have the A antigen, and type B, the B antigen. Now, maybe you're thinking, what is an antigen? An antigen is a marker on the outside of a cell, normally denotes something as belonging to itself or being foreign. We typically refer to antigens as ones that induce an immune response. But in this case, with red blood cells, like we're actually the self markers here, we're referring to them as antigens. And an antibody is something that will complement it. It's a Y-shaped protein produced by the immune system that will bind to it. It causes like grouping together or could pierce the cell as a defense mechanism against the pathogen. So the same applies here. If someone that is type B receives type A blood, then will they have the type A antibody? So that means that it's going to bind, it's going to cause blood clotting. And then, of course, a blood clot, if it got into your brain or if it got into your heart, is going to cause a stroke or a heart attack, and it's going to cause severe problems. So it's really important to know which blood type you're getting. There's like a funny transfusion game that you could easily Google and play, where you see if you can keep the patient alive and see if you give them the correct dose, like the correct blood grouping. There's something else called rhesus group, but we're not going to confuse ourselves with that today. You might have heard of it, so that's why I mention it. But typically for the IB, we're looking at the 4 blood groupings. Then there's type AB. So we call this individual the universal recipient. Why is it the universal recipient? Well, because it can ultimately receive A and B because it's producing neither antibody. Whereas type O would be the universal donor. It's able to donate this blood to anybody because it has no markers on it, so no antigens on the outside. However, it's now producing both antibodies, and that means it can only get type O. There is a classical cross that you could do where you could look at the inheritance. If you take an individual, now when we think about blood grouping, an interesting question would be like if you are AA or AO, that denotes blood group A. So the O would be where you don't have any antigen expressed for that. Sometimes given as a little I, and the A would be big I, big A, and I can show you that in a second. But what's interesting is that an individual that's AO and BO could have any 4 of the possibilities. So as I was saying, if you are blood group O, you have no antibodies on the outside. Make this one a little bit bigger again. And the classical question would be, well, what could the genotypes, could the parents be for us to get all 4 possibilities? So if you think about it, then if we are AO and BO in a cross, we can then get all four possibilities. So if I just insert a quick table and just show you what I mean, dun, 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 like this. Wasn't actually planning on doing this in the start. It's always good to be a bit organic, isn't it? And so below, thanks for bearing with me here. But like, so someone is AO, then there's A, and there's O as a possibility. And if someone is B, it could be B and O as a possibility. Don't need this column here. And then this individual is AB. This person is B. This person is A. And this person is O. So all four possibilities from that particular cross. So that's really interesting. Now, we're going to segue into talking about pedigree charts. Now, a pedigree chart looks a bit like this. And they look a bit horrible at first, but actually you're just always looking for this pattern. You might get a bigger pedigree chart. Like here's an exception one. So this is bigger, classic that you might get in a question. And you're trying to look for the patterns that you know. But what's actually happening here is it's mitochondrial DNA. So mitochondrial DNA is unique because you get that from your mother. We talk about inheritance as half from your mom, half from your dad, but ultimately what we're looking at is you get a little bit more from your mom because the whole idea of endosymbiosis, that mitochondria started off as a prokaryote that was engulfed and taken up by another larger prokaryote, and a symbiotic relationship was formed. And as a result of that, the mitochondrial DNA is not encoded in our within the nucleus of a cell. It's actually, or within those 23 pairs of chromosomes, this actually has its own DNA as a plasmid, and that's part of the evidence that mitochondria were originally prokaryotic cells, so endosymbiosis. Now, that means that you get that mitochondrial DNA from your mother. The mitochondria that in all of your cells all started off in her egg cell. That's an SL detail there, the endosymbiosis. But what that means is that if you have an affected mother from a mitochondrial disease, all of the offspring are affected. So circular denotes that it is a female, square means that it's male, filled in means that it has the condition, and blank means that it doesn't. And then these lines show the lineages. They have four offspring, all of them have the mitochondrial condition. Now, the mother will pass that on to all of their offspring. So here, kid number 5 had some kids, 13 and 14, they have the condition. But father doesn't pass on this mitochondrial DNA. So her 2 sons, 3 and 7, don't give anything off. Don't pass on the condition to their kids. That mitochondrial DNA came from their partner. Now, the other ones that we want to think about would be here with autosomal dominant. So 2 affected parents have an unaffected child. 2 unaffected parents have an affected child. So in the last video, we talked about Huntington's and cystic fibrosis, and it's the same 3-to-1, 2 heterozygous individuals here in a cross are producing this. And it's the same cross, however, this is an autosomal condition, this is a recessive condition, this is a dominant condition. And then we have the X-linked ones. And that's not something we're going to dive into today, I don't think. I think it's better to just stick with these two here, but I can introduce these and we can talk more about X-linked in another video. Right, you know what? I'll introduce the idea. So we're going to go and have a look at my web sheet. And up here we're summarizing codominance. And then I want to draw your attention to this idea of two affected parents have an unaffected child. And what's nice here is it pops up with the cross. And you can click on the other ones as well and see what their inheritance patterns are. And you can see what the parents are by clicking on, which I think makes it a bit nicer than the presentation. This is a skill as well. So any teachers watching, if you go to freeperiod.ai, you can check out the skills that I make for Claude, and you can turn any lesson into one of these interactive HTMLs. And students, you could do that too. If you've got a slide deck from your teachers, you could equally do that. It's quite nice to review. And this particular skill will give you some practice IB questions as well. So here we've got 2 affected parents have an unaffected child. Always look for these rules when you're doing a pedigree chart. 2 unaffected parents have an affected child. Same cross, but the inheritance is on the recessive. So here, the inheritance would be here, like the dominant gene is that's the ones that have it. This individual, homozygous recessive, doesn't have the condition. Now, when we think about X-linked, we're talking about the X and Y chromosome. The X chromosome is longer than the Y chromosome. So there is no competing gene, like allele of a gene, in order to like mask the effect of the other one. So the X is much longer and the Y is much smaller. And then therefore there is no allele of that gene on the Y chromosome sometimes that could mask the effect, such as male pattern baldness, something that I can strongly relate to. I blame early teaching and the stress of managing 32 kids in a British state school. So that's why I've sold out and ended up in Switzerland, amongst other reasons, like I love snowboarding and skiing, but I'm digressing. Now, here when it's an X-linked, I'm going to click on this, is an affected male. It's an X-linked dominant condition, and you can see has all affected females. So we look at the cross here, and the male is passing on his X chromosome to daughters. Because the male is actually determining whether or not an individual is male or female because it's providing the X or the Y chromosome. Now, if we look at an affected mother, has all affected sons. So this could be like color blindness or hemophilia, and these are the typical examples. Now, because we're getting the X chromosome, the sons are getting the X chromosome from their mother, the mother is determining whether or not they have an X-linked recessive condition in this instance here. Now, these are just 2 rules. Of course, you can't apply them all the time, but within the context of IB, there's going to be one of these 4 rules present. Maybe the exceptional one with the mitochondrial DNA. I've not seen that question yet, but it's now included in the syllabus. So it doesn't mean it won't be there. So if you know those 5 rules, then you can figure out the answer to one of the pedigree charts. So it's a really nice, I think, question to be getting in a multiple choice exam. Just flicking through, showing you that at the end of these web sheets, we've got some nice practice questions here that could help prepare you for an exam. So I'll drop the link to that below. Don't forget to like, share, and subscribe. Thanks for another episode. And stay tuned for the next one. I'm taking any requests, so if you make a comment, I'm happy to do a video on something that you got coming up in class, or something that you have some confusion or misconceptions around. All right, ciao for now.